The DNA strand is antiparallel. It replicates in the 3 to 5' direction.
Helicase is an enzyme. It breaks the hydrogen bonds between the bases of the parental strands. Single-stranded DNA binding proteins stabilize the now separated DNA strands, keeping it in an extended single-stranded state.
DNA polymerase, accompanied by a sliding clamp, can now copy the DNA. DNA polymerase copies the nucleotides, while the sliding clamp holds the polymerase in place.
The new strand formed from the leading strand template is formed in the 5 to 3' direction. This is known as the leading strand.
As the DNA polymerase and sliding clamp move along the leading strand template, these single-stranded DNA binding proteins are removed as they are no longer needed.
RNA primer and RNA primase are now required for replication. Primase creates short RNA segments called primers. RNA primers allow polymerase to initiate synthesis of the new strand.
Short segments called Okazaki fragments are formed from the lagging strand template. These segments collectively form the lagging strand.
The clamp-loading protein associates both DNA polymerase molecules in opposite parental strands. It is bound to helicase. These associations made by the clamp-loading protein coordinates the simultaneous synthesis of both DNA strands at the fork.
The leading strand is continuously synthesized in the direction of the replication fork. The lagging strand template must be folded so its polymerase can create DNA in the 5 to 3' direction and still move in the same direction as the overall movement of the fork.
Using the 3' end of the primer, DNA polymerase lays down the new DNA in the 5 to 3' direction. The DNA polymerase then dissociates from the DNA, but remains attached to the clamp-loading protein. The clamp-loading protein places a new sliding clamp and places the DNA polymerase at the next primer. The process repeats.
As DNA polymerase has replaced the RNA primer, finally DNA ligase can link the Okazaki fragments. The lagging strand is now formed, and replication is complete.